I am Mike Arnold. I'm an evolutionary biologist. I work at the University of Georgia in the Department of Genetics. I've worked on a number of different kinds of organismic groups, including both plants, animals, and also fungi, which we consider to be plants or animals sometimes. And I'm very interested in how gene movement between lineages, divergent lineages of organisms, how that might affect evolutionary history, evolutionary trajectories, ecological trajectories of organisms. Reticulation or reticulate evolution, a number of us have typified this with a metaphor sort of in contrast to the tree of life. We talk about a web of life, and reticulation and reticulate evolution is the reflection of the observation that as organisms diverge from a common ancestor, they often encounter one another and hybridize or have horizontal gene transfer if they're prokaryotes, or viral recombination if they're viruses. And this leads to what's referred to as a reticulate pattern of evolution, a web, sort of like a spider web. Is how we picture it sometimes, where the connections between the different strands reflect gene exchange or recombination of the genomes of the divergent organisms. One of the really fascinating parts of reticulate evolution, I think, is that it impacts our own species. It has impacted the evolutionary history of our own species. So many different pieces of information from our own genome, sequences from populations of our species scattered across the world, indicate that there are genes there that were introduced from introgressive hybridization or natural hybridization between our species and what are referred to as archaic species, those that are extinct but belong to our genus, say Homo neanderthalensis, or there's another form called Denisovans that it looks like we hybridized with. And so our genome that we have now has genes from those other species from our genus. So that's one reason it's very important for us to understand reticulate evolution, because we are, in general, because we're understanding our own evolutionary history. But reticulate evolution also impacts us in our ecology, those in our situation and the organisms we interact with. If you're wearing cotton right now, which most of us do, the reason we can have cotton clothes is because, it's estimated about 10 million years ago, two very divergent kinds of plants hybridized, mated, and formed this new form that we came along about 10 million years later and started selecting for a plant that produced long fibers and nice fibers. Without that hybridization event, we now know the genes would not have been put together that allow us to use that product and make many, many clothes out of. But it also, so that's a benefit to us from reticulate evolution, but it's also, there are detrimental or even catastrophic factors that happen to us as a species because of reticulate evolution. For example, this year, in 2013, 80% of, it's estimated, about 80% of the people that had immunizations for influenza, at least in North America, and I believe this was worldwide, 80% of those folks who had the shot still caught influenza. And the reason is because there was viral recombination in between the time the World Health Organization and other health organizations predicted what influenzas were going to be circulating in our population. They predicted a certain set of them, and they guessed wrong. It wasn't their fault, they weren't unintelligent, it was just that there was an event where two viruses or more recombined, ending up with a new variant that they didn't predict and didn't put into the immunization scheme. So, disease-wise as well, reticulate evolution can have a very detrimental effect on our populations. During the neo-Darwinian or modern synthesis is what we refer to it, either one of those terms. During the modern synthesis, which ran, everybody probably knows this, but it ran from say the 1930s, maybe through the end of the 1950s, something like that. Gets a little vague on the edges there. During that time, there were definitions given to species. There was definition given to conceptual framework built up around how species evolved. And that was central to the idea of how evolution, the evolutionary process occurred. Now the modern synthesis, what they were referring to there as well, was that they took the field of genetics, and that was the anchor point, if you will, for understanding then how species related to one another, how species came about. You know, what kind of genetic changes might lead to speciation, process of species formation. In particular, one of the things that they argued during the modern synthesis was that species are reproductively isolated, completely reproductively isolated from one another. They cannot mate, or they're not different species. And that species arise in isolation, geographic isolation, it's called allopatry, from one another. Populations in allopatry diverge genetically, they diverge in terms of adaptations, and then if they encounter one another again, if their ranges expand, they will not reproduce if they're different species, or will not reproduce with one another if they're different species. What the extended synthesis and since the modern synthesis, and in particularly the last 20 years, what we've come to understand, a number of labs around the world, including my own, have come to understand is that in actual fact, it appears that species do not arise in isolation from other species, complete isolation anyway, and that there is some genetic exchange as a general process, introgressive hybridization, horizontal gene transfer in bacteria's viral recombination, as those lineages diverge, and that natural selection still selects and pushes them apart in one sense, selecting for different phenotypes. But the modern synthesis and the neo-Darwinian synthesis really had that wrong in terms of how species have to be in complete isolation, or populations in complete isolation to diverge into different species. That was the concept, and now we understand that that's probably incorrect for many of the speciation events. One of the aspects of my work that's been fun is been very enjoyable is to try to synthesize large pieces of data, or large data sets I should say, from ecology and evolutionary biology, from developmental biology, whatever it happens to be, and to test with that comparative approach whether or not reticulate evolution has been important for organisms in general. And one of the ways that I've synthesized that, or put out a product of synthesis, is to write some books. And the one that we're using in the course here in Lisbon is called Evolution Through Genetic Exchange. It came out in 2006, and I think it's a nice synthesis for students, but it's also a really good tool for me to think about the concepts, the mechanisms, the methodologies that we would use to test the outcome, for example, of the evolutionary process in general, but also the reticulate evolutionary process.